Mitosis and meiosis are two of the most important processes in all of biology, and understanding the differences between them is key for both exams and real biological insight. Mitosis is used for growth, repair, and replacing old cells, while meiosis is used specifically to create gametes for sexual reproduction. Although both processes involve chromosome replication and separation, the goals and outcomes are entirely different. Students often mix these processes up because they share names for their phases, but the events occurring within the phases are very different. This video will break down both processes clearly and visually so you can finally keep them straight. And yes, there'll just be a hint of gentle humor to make chromosome behavior slightly more pleasant. Before we dive into the detailed stages of mitosis and meiosis, it helps to understand why cells divide in the first place. Mitosis is essential for everyday life. You grow from a single cell by mitosis, and you repair tissues by mitosis when you get a cut or bruise. This process makes exact genetic copies, which is important so your skin cells don't suddenly decide to become liver cells. Meiosis, however, is an entirely different story because it creates gametes for sexual reproduction. These cells need to have half the normal chromosome number so that fertilization can restore the diploid state. Meiosis also reshuffles genetic information, creating the diversity that allows populations to adapt, and giving future biology students lots to memorize. Chromosome vocabulary is where many students get tripped up before they even start learning the phases. A chromosome is a single DNA molecule packaged with protein, but once it has been replicated, it consists of two identical sister chromatids. These chromatids stay attached at the centromere until pulled apart later. Homologous chromosomes, on the other hand, are a pair of chromosomes you get from your parents, same genes, but not necessarily the same versions of those genes. Distinguishing between sister chromatids and homologous chromosomes is critical when comparing mitosis and meiosis. Once these definitions are clear, the two processes make much more sense, and diagrams stop looking like abstract noodle art. Mitosis is a straightforward division that focuses on making two genetically identical cells. Before mitosis begins, the DNA is replicated, so each chromosome has two sister chromatids. In prophase, chromosomes condense and spindle fibers form. Metaphase lines the chromosomes up across the middle of the cell, like students taking assigned seats. Anaphase pulls sister chromatids apart, ensuring each side gets the same genetic information. Finally, telophase and cytokinesis form two identical daughter cells, ready to carry out normal cellular functions. No homologs pair up in mitosis. It's a very orderly, drama-free event compared to meiosis. Meiosis is more complex than mitosis because the goal is to produce gametes with half the normal chromosome number. The process is split into two rounds of division, meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes pair and exchange genetic segments in a process called crossing over. This recombination, along with independent assortment, ensures that every gamete is genetically unique. Meiosis II resembles mitosis, but separates sister chromatids rather than homologous chromosomes. By the end, four genetically distinct haploid cells are produced. Quite an achievement for one original diploid cell. The most important distinction between mitosis and meiosis occurs during meiosis I. In this phase, homologous chromosomes pair up in a process called synapsis and then separate into distinct cells. This pairing never happens in mitosis, which deals only with sister chromatids. Crossing over, which takes place during prophase I, exchanges DNA between homologous chromosomes and greatly increases genetic variation. Because homologs separate in meiosis I, the chromosome number is reduced from diploid to haploid. This reduction does not occur in mitosis, which maintains chromosome number exactly. While meiosis I is dramatically different from mitosis, meiosis II looks surprisingly similar to it. In both processes, sister chromatids line up along the metaphase plate and are pulled apart by spindle fibers during anaphase. There is no pairing of homologous chromosomes and no crossing over occurs. One major distinction is that meiosis II begins with haploid cells, while mitosis begins with a diploid cell. Despite their similarities, meiosis II still contributes to the overall genetic variation produced by the whole process. Each chromatid separated in meiosis II may already be recombinant because of earlier crossing over. Meiosis is the powerhouse of genetic variation in sexually reproducing species. Crossing over swaps genetic material between homologous chromosomes during prophase I, creating entirely new allele combinations. Independent assortment adds another layer of randomness by shuffling the homologous chromosome pairs during metaphase I. The result is a collection of gametes that are all genetically unique. Once fertilization occurs, yet another source of variation is introduced. Which two gametes combine is entirely random. Mitosis cannot compete in this department, as it produces genetically identical cells unless a mutation sneaks in. When comparing the outcomes of mitosis and meiosis, the differences become especially clear. Mitosis ends with two identical diploid daughter cells, each capable of functioning just like the original parent cell. Meiosis, however, ends with four genetically distinct haploid cells that will become gametes. These different outcomes reflect their different purposes. Mitosis focuses on maintenance, while meiosis focuses on reproduction. Chromosome number remains constant throughout mitosis, but is halved in meiosis. These distinctions frequently appear on exams, so it's worth memorizing them as clearly as possible. Meiosis is usually a well-orchestrated process, but like any complex event, things can occasionally go wrong. Nondisjunction occurs when chromosomes fail to separate properly during meiosis I or meiosis II. This mistake results in gametes with too many or too few chromosomes. If such a gamete is involved in fertilization, the resulting zygote may have a chromosomal disorder such as Down syndrome or Turner syndrome. Non-disjunction becomes more likely with increasing maternal age due to accumulated cellular stress on oocytes. While chromosome segregation errors can also occur in mitosis, they are far less common and usually associated with abnormal cell growth. The differences between mitosis and meiosis become easier to remember when viewed side by side. Mitosis produces identical cells through a single division, while meiosis produces genetically varied gametes through two divisions. Only meiosis involves crossing over and independent assortment, which together create extraordinary genetic diversity. Homologous chromosome pairing is a hallmark of meiosis 1 and never occurs in mitosis. Chromosome number remains constant in mitosis, but is reduced in meiosis to ensure proper fertilization. Together, these two processes maintain life's balance between stability and diversity. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.